This is a video lecture on Hybridoma technology for the production of monoclonal antibodies. Myself, Professor Sujata Mahapatra from the Department of Botany, Rama Devi Women's University, Bhubaneswar. Hybridoma technology is a part of the course curriculum of MSc Industrial Microbiology, MSc Biotechnology, and MSc Life Sciences. During the discussion, there will be mention of these terms: antigen, antibody, epitope, paratope, plasma cells, antibody clone, monoclonal antibody, and myeloma cells. Antigens are macromolecules which may be a protein or polysaccharide or their conjugates, and these induce immune response. Antibody are the proteins which belong to the immunoglobulin family, and they are produced in response to the antigen. When the antigen enters into the body, then the immune system gets triggered and antibody are produced. There are 5 classes of antibodies depending upon their structure. Epitopes are molecular sites which are present on the antigen. Multiple epitopes are always present, and multiple epitope, they induce the formation of multiple types of antibodies. Paratope is the region on the antibody that interacts with the epitope on the surface of the antigen. Plasma cells are the B lymphocytes which are capable of producing antibodies. Clone of antibody means a group of antibodies produced from a single plasma cell. Monoclonal antibody is a clone of antibody induced against only a single epitope on the surface of the antigen. Myeloma cells are cancerous plasma cells. Hybridoma technology. It is a method of production of monoclonal antibodies. Hybridoma means a hybrid is created first, and this specific hybrid cell produces or secretes monoclonal antibodies. This method was first standardized by George Kohler and Cesar Milstein in the year 1975, and for their significant contribution, these two scientists, along with Niels John, received Nobel Prize in Physiology or Medicine in the year 1984. The nomenclature of antibody is according to certain principles, and it is recognized by INN and USAN. This is the picture of an antibody molecule. Each antibody molecule has some specific regions. There are 2 light chains and 2 heavy chains. On the basis of molecular weight, they are categorized as light chains and heavy chains. Light chain has a variable region and a constant region. Heavy chain has 1 variable region and 3 constant regions. Inside the variable region of the both light chain and the heavy chain, certain groups are there which actually interact with the antigens, or specifically they interact with the paratope, and these are called as hypervariable regions. Now the difference between a monoclonal and a polyclonal antibody. This is a general picture of a Y-shaped antibody molecule, and this shows an antigen molecule with a single epitope. When it interacts with the immune cells, single epitope results in the secretion of only one type of antibody by the plasma cells. This is the antigen molecule with multiple epitopes on the surface, and multiple epitope induce the production of different types of antibodies. So when the antibody variety is more, it is called as polyclonal antibody, and when only a single type of antibody are produced, these are called as monoclonal antibodies. At the beginning of this production technology, only mouse MAB was produced and a hybrid was created by fusing the spleen cells of the immunized mouse with the myeloma cells of the mouse. So it is totally mouse-based hybrid cell, and this hybrid produces the monoclonal antibody. It has its side effects. When injected into the human, there is immunological reaction as it is completely different from the human system. Second type of monoclonal antibody is chimeric monoclonal antibody. It is produced by fusing the variable region from the mouse immunoglobulin gene and the constant region from the human, and this DNA, which is produced by the fusing these two regions, gives rise to the antibody, monoclonal antibody, after proper procedure. The third type of monoclonal antibody is humanized antibody. Humanized means it is not completely human based. Some region belong to the mouse. The complementary determining region or the hypervariable region belongs to the mouse, and the other portions, most of the portions, come from the human. And daclizumab is an example of humanized monoclonal antibody. Then fully human monoclonal antibody. These are produced by injecting the human immunoglobulin genes into the mouse. The transgenic mice, which are genetically engineered in this way, they secrete the fully human monoclonal antibody, and they are non-reactive, and because there is no mouse derived proteins when it is injected into the human body. Bispecific antibody, this is the example. Two hybridoma are created against different types of antigens, and the hybridoma cell produces two types of antibodies. Because there are two hybridoma, they will produce two types of monoclonal antibodies, and when these two antibodies are fused, we get bispecific antibodies with two different antigen interacting arms. These are the different types of monoclonal antibody as discussed in the previous slide. But here there are examples of the mouse-derived monoclonal antibody, humanized monoclonal antibody, then human monoclonal antibody, etc. There are different steps of the monoclonal antibody production. The basic thing is two types of cells are required. One is the plasma cell and one is myeloma cell. Myeloma cells are cancerous plasma cells. So plasma cells and myeloma cells are fused. Plasma cells have the property to secrete the antibody. Myeloma cells do not have the property to secrete the antibody, but they have immortality. So due to the immortality feature, and this is for the antibody secreting feature, both are combined to produce a hybrid cell that is called as hybridoma. And hybridoma is capable of secreting monoclonal antibody. This is the overview of the procedure. Two types of cells are required. One is the spleen cell, that is the source of plasma cells from the immunized mouse. Second is the myeloma cells. The plasma cells are HGPRT positive and immunoglobulin positive. They have functional HGPRT gene and they can secrete the antibodies. So mouse is injected with a particular antigen. Then after a few days and some screening, the splenocytes are removed, and from the splenocyte the plasma cells are collected. Myeloma cells are specific mutant cells which are HGPRT negative and immunoglobulin negative. So once myeloma cells and the plasma cells are obtained, they are fused in the presence of polyethylene glycol or by electrofusion. After fusion, there is a possibility of unfused B cell, plasma cell, unfused myeloma cell, and different types of hybrid cells. Now the hybrid cells are grown in a hat medium, that is a selective medium that contains three chemicals: hypoxanthine, aminopterin and thymidine. And in this hat medium, the B cell will die, myeloma cells will die, only the hybrid cells will survive. The hybrid cells are further screened to get the desired hybrid cell. The desired hybrid cell is cultured in culture plates. After that, from the wells, the hybrid cells are picked up and cultured in conical flask in a selective medium. And for in vitro method, the hybrid cells are injected into the mouse. After some days, the mouse will secrete the monoclonal antibodies. The first step of hybridoma technology is the collection of plasma cell and the myeloma cell. Plasma cells are obtained from the mouse, and mouse is injected either with only the antigen for which MAB is going to be produced, or the antigen may be conjugated with an adjuvant. Adjuvant is a small molecule which will induce a better immune response, and this method is repeated at some interval. Then from time to time the blood sample of the mouse is drawn and it is tested through ELISA or flow cytometry to detect the level of antibody titer or the concentration of the antibody. Once the concentration reaches a specific level, the mouse is sacrificed and the spleen is removed. Spleen is subjected to density gradient centrifugation, and the splenocytes will release the activated plasma cells, and these plasma cells are collected. The plasma cells are capable of secreting the antibodies. These are HGPRT positive and immunoglobulin positive, means they can produce immunoglobulin and they have a functional HGPRT gene. Myeloma cells are B lymphocyte cancer cells and they have certain properties. For the hybridoma technology, myeloma cells are selected because they have immortality. The myeloma cells for this particular procedure must be HGPRT negative and they are mutated myeloma cells. So what is this HGPRT gene? This is the gene that is hypoxanthine guanine phosphoribosyl transferase. The nucleotide biosynthesis involves the de novo pathway. Under certain situation, the de novo pathway cannot operate, and under that circumstance, there is a salvage pathway or alternative pathway of nucleotide synthesis. This enzyme, it is required for carrying out the salvage pathway. If this enzyme is not functional or it gives rise to a defective product, then the cell cannot carry out the salvage pathway. So these myeloma cells are the cells which are HGPRT negative. These myeloma cells before fusion are cultured in the presence of 8-aja guanine, and the 8-aja guanine is usually cytotoxic to normal cells, but the mutant myeloma cells are HGPRT negative and they are not killed by the exposure to 8-aja guanine, or the myeloma cells which are HGPRT negative are resistant to the cytotoxin 8-aja guanine, which is a purine derivative. So the myeloma cells, when they are grown in presence of the 8-aja guanine compound, the cells which are resistant are collected. The resistant cell must be HGPRT negative, and for the procedure of the fusion, HGPRT negative myeloma cells are required. So these mutated myeloma cells are collected from the culture medium. Third step is the fusion of plasma cells and the myeloma cells. There are different methods of fusion like the electrofusion or the PEG-mediated chemical fusion. So this diagram shows the electrofusion. Alternatively, the myeloma cells and the plasma cells can be grown in the presence of the cell-fusing agent for polyethylene glycol that will induce the fusion of the plasma membranes and that will produce the hybrid. Fourth step is selection of a hybrid cell or screening for a hybrid cell. After the fusion procedure is over, the hybrid cells are cultured in a selective medium that is called as HAT medium. HAT because it is composed of three chemicals: hypoxanthine, aminopterin, and thymidine. Aminopterin blocks the de novo pathway, so the cells which cannot carry out the salvage pathway will not survive in the HAT medium. So in our procedure, there are two types of cells, the myeloma cells and the plasma cells. Plasma cells are HGPRT positive and they can carry out the salvage pathway. So even if aminopterin blocks the de novo pathway, the plasma cells will survive by carrying out nucleotide biosynthesis by the salvage pathway because they are HGPRT positive. But the B cells, which are HGPRT positive, do not have the power to survive long term. They do not have immortality. After some time, the B cells will automatically, they will die because they cannot divide permanently. They are not immortal. Now, unfused myeloma cells. These are HGPRT negative. They cannot carry out the de novo pathway because of the presence of aminopterin, and as they are HGPRT negative, they cannot carry the salvage pathway. So these mutant myeloma cells which cannot adopt either pathway will not survive. So unfused myeloma cells, they die because they are HGPRT negative and the selective medium contains aminopterin which do not allow the de novo pathway. B cells die because they have limited lifespan. Only the hybrid cells which are HGPRT positive as well as they have immortality from the myeloma cells, they will survive in the HAT medium. There are different types of hybrid cells and these hybrids are screened to collect the required hybrid from different types of hybrid cells. The hybrid cells are screened because there are different types of hybrid cells. The hybrid cell which produces the specific monoclonal antibody has to be selected. This is done by using different immunotechniques like radioimmunoassay or immunofluorescence. After selecting a particular hybridoma, these are cultured in culture plate in vitro method. In vitro method, from the culture plate, the hybrid cells are transferred to flasks and again cultured till a particular cell density is achieved, and these hybridoma cells then are used to collect the monoclonal antibody. In vivo method, the few hybridoma are injected into the peritoneum of the mouse, and after a few weeks, the ascites of the mouse are collected to get the monoclonal antibody. The in vitro method gives purer monoclonal antibody as compared to in vivo method, because in vivo method, the mouse proteins may be contaminating the monoclonal antibody. These are the applications of MAB: cancer diagnostic imaging, diagnosis of infectious diseases, treatment of infectious diseases, induction therapy in transplantation, and cancer therapy. MAB can be used for cancer treatment, and as per this link, 24 MABs have been approved for cancer treatment till now. The MAB, the advantage is that any cancer drug which is used, mostly the chemotherapeutic agents, have severe side effects and they are very harmful to the recipient, the patient. But the MAB is produced against a particular cancer antigen, and if one MAB is directed to this type of antigen, then it will touch only the cancer cell. It will not interact with any healthy cell. So that is why when the MAB is directed to only the cancer cells, only the cancer cell will be killed by the drug carried by the MAB. So this is called as drug targeting by the MAB, and the rituximab is an approved and the first ever MAB that was used for cancer in 1997, and it is being used till now for chronic lymphocytic leukemia. Cetuximab is also used for advanced bowel cancer. So what is the mechanism by which MAB acts on the cancer cell? There are several mechanisms. One is MAB binds to the oncoprotein that is produced by cancer cells, and the binding between the cancer antigen or the oncoprotein and the MAB triggers the immune system. Cancer cells have a quality to evade the immune system, but when MAB binds to the oncoprotein, it triggers the immune system by inducing signal transduction pathways that will alert the immune system, and then the immune system cells will be targeting the cancer cell and trying to eliminate the cancer cells from the body. Some MABs, they directly interfere with the growth of the cancer cells because they inhibit the cell cycle checkpoints and the cancer cell cannot divide and grow. Another mechanism by which the MAB is beneficial, MAB surrounds the cancer cell. It produces a coating around the cancer cells, and these coated cancer cells are recognized by the immune system and they are destroyed by the immune cell. Cancer cells survive in different manners, different ways. One method of survival is they prevent the activity of the immune system. So they prevent the activity means they produce certain proteins, and these proteins interfere with the immune system and block the access of the immune cells to the cancer cells. So these MABs, they directly get attached to the proteins and stop the action of these proteins so that cancer cells are attacked by the immune cells and there is a chance of killing of the cancer cells. Another way by which the MAB treats the cancer cells or it removes the cancer cell is it blocks the signal for angiogenesis. Angiogenesis is the production of blood vessel by the cancer cells. The cancer cells produce abnormal types of blood vessels to collect nutrients from the healthy cells and the tissues. And the MAB, certain types of MAB block this signal for angiogenesis, so the cancer cell cannot carry out angiogenesis. These type of MABs are called as angiogenic drugs. Cancer imaging also uses monoclonal antibody. So here there are mention of two examples of monoclonal antibody that have been approved for the detection of recurrence of colorectal cancer and the small cell lung cancer. In this technique, the MAB is first produced against the tumor antigen, then it is radiolabeled. The radiolabeled MAB is injected into the body of the patient. It binds to the particular cancer cell, and after this application is followed by imaging by using SPECT and the immunoscintigraphy. Then the location of the cancer cell can be detected in the body by this non-invasive method. Organ transplants are very risky because the immune system of the recipient may reject the transplanted organ, and immunosuppressive drugs are given to prevent the rejection of the transplant by the patient's body. Monoclonal antibody can be used both before and after organ transplantation. They are immunosuppressive by nature and they prevent allograft rejection. At least they reduce the chance of allograft rejection by the immune system. This type of therapy is called as induction therapy, and it increases the tolerance to allografts. And these are two MABs which are used in case of kidney transplant patient to prevent the rejection of the transplanted kidney. Tumor markers are molecules produced by the cancer cells, and if a specific MAB is getting produced against a tumor marker, it can be used for immunodiagnostic method for the cancer detection. And there are different examples of MAB which are produced against carcinoembryonic antigen in case of colon carcinomas. MAB can also be used in the identification of the pathogen and the treatment of pathogen-induced diseases, both in case of humans and animals. So, MABs, which are developed against specific antigen from the pathogen, can be used in immunotechniques like ELISA, Western immunoblotting, then radioimmunoassay, fluorescence microscopy, etc. for the diagnosis of specific bacterial, fungal, viral or parasitic diseases. MAB, which is produced against exotoxin of certain bacteria, have been approved for the treatment by FDA. These are called as antitoxin MABs. They limit the virulence and recurrence of the infection. Also, they may reduce the pathogen invasion chance. These are the examples of MAB which are either in research state or they have been approved. This diagram shows how the process is done. First, this is cancer. It has different antigens. For a particular cancer antigen, MAB is created. Then it is radiolabeled. Radiolabeled MAB is injected into the body. And if there is recurrence of cancer at a particular location or there is metastasis, then the MAB will bind to that particular cell, and this is followed by the imaging techniques that will show where the MAB has conjugated. So that is the place where the cancer cells are present. These are some other uses of MAB. They can be used for the migraine prophylaxis or prevention of the migraine attack, for rheumatoid arthritis therapy, plague psoriasis, Crohn's disease, for the treatment of autoimmune disorders like SLE, system lupus erythematosus, that is an autoimmune disorder, and MAB have been produced to treat this type of autoimmune disorders. Mouse MAB may not be tolerated by the human system and it may cause severe allergy reaction. So currently, the immunodeficient mice are used for in vivo production of MAB. In vitro production methods are also used. Mass production of MAB can be done by using bioprocessing techniques. In vivo method is the choice for diagnostic use and in vitro production for therapeutic use. MAB are very important for human as well as livestock healthcare, but it is not without its side effects. Mostly, the application of the mouse MAB, it induces different types of reactions like low blood pressure, diarrhea, fever, fatigue, and acute allergy reaction of the whole body, that is anaphylaxis. So if there is more research in this area, one day MABs may be produced which are tolerated by the human system, and then they will be really useful in medicine and healthcare. Thank you.